Good afternoon, everybody. My name is Gilles Salle from Memorial Sloan Kettering, and I'm delighted to be part of these organizers that led to this meeting and opened the second session. I wish I could be with you in London, but for administrative reasons, I had to stay where I work. Having said that, we have an exciting session coming up, which I share with my colleague Chiara Bonini. And it's really my honor to introduce the first speaker, Professor Malcolm Brenner from the Center for Cell and Gene Therapy at the Baylor College of Medicine, founding director of this center, affiliated with Houston Methodist Hospital and Texas Children's Hospital, and Fayez Sarofian, Distinguished Service Professor at Baylor College. I don't think Professor Brenner needs a long introduction. He trained in United Kingdom at the University of Cambridge and obtained his medical and PhD degrees and moved in the early 90s in the US. And I think many of us may remember the first presentation of gene therapy within gene transfer into stem cell. And he was the first to perform those outside of NIH and the first to use marrow stem cell as a target. He moved in 1988 to his current position, and over the last 30 years, he has devoted his career as a physician scientist to the field of stem cell transplantation and therapeutic use of T-cell immunologic approach and genetic engineering strategies. He serves as the editor-in-chief of Molecular Therapy, former president of the ISGCT and the International Society of Cell Therapy. I think he earned a widespread recognition and many awards, and he has been elected as a member of the National Academy of Medicine. Today, he will address a keynote lecture providing us an overview of cell therapy for cancer in 2022 and beyond. Professor Brenner, the floor is yours.
Thank you very much. And as others have said, it's a pleasure finally to be here and present in person. These are my disclosures. I'm grateful to the organizers, not just for inviting me and for that introduction, but also for giving me such a nice broad title. I'm going to narrow it down a bit, otherwise it'll be beyond the limits rather than at them. And I'm going to talk about immune effector cell therapy, and in particular, whether solid tumors are actually in reach of these therapies. And I'm focusing on that because I think events in biotech over the past few months have made it very clear that we're not going to get the inward investment and interest that the field needs to innovate and execute unless and until we can show that we can not only treat but even cure at least some solid tumors in the same way that we can treat and cure at least some hematologic malignancies. But of course, to do that, we need to overcome a series of obstacles. Not least, we need to increase accessibility in the broadest sense of the word. There are probably 8 or 10 times as many solid tumors as there are hematologic malignancies, and we struggle even to cope with those at the moment. We need to target tumor-specific antigens rather than lineage-restricted antigens because, of course, most solid tumors arise from organs whose lineage is not dispensable. And we need to acknowledge that solid tumors in humans are very heterogeneous in expression of these tumor-specific antigens and are very plastic. It changes over time. And we also need to overcome the inhibitory and highly reactive responsive tumor microenvironment, which again changes as the immune attack changes. Now, many of the speakers will, and in some cases already have, deal with all those issues in much greater depth and detail than I'm going to. But what I wanted to try and do was set an overall context and illustrate some of the ways we're trying to address these problems from very early phase clinical studies with very preliminary data. Okay, increased accessibility. So for autologous products, I think accelerated and also automated manufacturing, potentially ending up with point-of-care preparation and administration, I think that will help a great deal. But I also believe that for solid tumors, we're going to need to rely for the heavy lifting on allogeneic or off-the-shelf products. And the reasons for this are not just that they are even more immediately available and should have lower cost of goods, but because they have a much more predictable potency, or should have. And that is going to be an increasing issue dealing with solid tumors which occur in a generally older population for the ones that we treat, and in whom there's much greater variability in the tumor and the tumor treatment. And having a product at least that is reasonably consistent may make the design and analysis of clinical trials somewhat easier. Two limitations of off-the-shelf cells, as we know, are that the host rejects the therapeutic product, or that the therapeutic product rejects the host, so-called graft versus host disease. In immunocompetent individuals, it's rejection that dominates. In the immunodeficient, it's graft versus host disease. So starting with the rejection issue, I think it's probably fair to say that most people are focusing on shielding the off-the-shelf product from immune attack when it goes into the recipient, for example, by removing HLA molecules. And certainly this has an impressive benefit. The problem is that defenses against allogeneic cells are very deep. They're not just alloreactive T cells, they're NK cells and many other cellular and humoral mechanisms. And the polymorphisms that are recognized are equally broad. And simply eradicating one or two of them probably won't be enough for long-term engraftment. So we've taken a slightly more active approach, which relies on the fact that as cells become activated, immune cells become activated, they express on their surface a range of new receptors and ligands. And if we could make the off-the-shelf immune effector recognize those new products, we could attack the alloreactive cells and destroy them before the product was itself damaged. And we focused on looking at a lot of different members of the tumor necrosis factor receptor family, and I'm going to talk about two of those, 4-1BB and CD30, beginning with 4-1BB. So 4-1BB pretty much follows the standard pattern of these molecules, with increased expression on activation or alloactivation declining over a few days. For 4-1BB, it's greater on CD8s than on CD4s. IMO developed an alloimmune defense CAR, an ADCAR, that was made up of a 4-1BB modified ligand and a CD3 zeta signaling domain. And she showed that this gave cytotoxic function to the off-the-shelf T-cell product and allowed it to destroy activated alloreactive T cells and NK cells as well. And we can get very high levels of expression of this ADCAR, 95 to 98%, and the cells that express the ADCAR grow very well, just as cells with any other CAR expression grow. So there's no evidence of fratricide, something we can perhaps come back to in the question period. Importantly, they act in the way that we want them to. So they don't do anything to resting allogeneic cells, but if we alloactivate those cells, they destroy both CD4 and CD8 alloreactive cells, more CD8 than CD4 because of the differential expression. In terms of using them as a means of getting alloengraftment of a CAR, we can co-express both solid tumor-directed and hematologic malignancy-directed CARs at a very high level, so that about 95% of the cells are dual expressors. And these cells behave in the way that you would want them to behave, at least in an animal model of solid tumors, various solid tumors, and also of heme malignancies. I'm just showing NALM6, B-cell malignancy, where we see in a xenograft model in NSG mice that the leukemia engrafts rapidly and kills the mice rapidly. If we give those mice CD19 CAR T cells from an allogeneic standard human donor, the tumor growth is controlled and the mice are long-term survivors. But if we first engraft those mice with a different set of T cells from a different donor, then the CAR T cells are rejected by this engrafted human immune system and the mice die. Only if the off-the-shelf product expresses both the therapeutic CAR and the ADCAR do we get recontrol of the leukemia or the solid tumor and mouse survival. So we've gone forward to test this approach in humans, not with 4-1BB, but with CD30. CD30 is expressed in the same kind of way as 4-1BB, going up on activation or alloactivation, but is about equivalent in CD4s and CD8 cells. And the reason for choosing this was that it's very highly expressed by many Hodgkin lymphomas and some T-cell lymphomas, and we can actually treat these lymphomas with autologous CD30 CAR T-cells. In other words, in principle and potentially, CD30 targeting CARs could be both a therapeutic CAR and an alloimmune defense CAR, an AD CAR. So just to show what I mean, I'll show the results from our autologous study by Barbara Savaldo and Carlos Ramos. It's a two-center study at UNC, University of North Carolina and Baylor. Cytoreduction prior to CAR T infusion, the standard flu-cy, infused 0.5 to 2 times 10 to the 8, and 28 subjects with multiply relapsed disease. These patients had a median of 5 prior lines of therapy, including current immunotherapies. The cells expand in vivo very nicely, looking like most other CARs in a dose-dependent manner, and they are very effective. About 66%, 18 of 27 patients in this two-center study had a complete response, and about a 50% one-year disease-free survival. Tessa Therapeutics have taken this on as a pivotal study, and in the pilot phase of that, again multi-center, they've obtained broadly comparable results, and they're now going into the full-scale study. Well, since CD30s can target tumor, we needed to find out if they could also prevent rejection when they were used not in an autologous setting, but in an allogeneic one. This doesn't, of course, address the issue of graft versus host disease, which is a consideration because these patients are going to be somewhat immunocompromised, not least by their prior therapy and by the flu-cy conditioning. 3 major ways of avoiding GVHD. You've heard of one of them, which is to remove the responsible TCRs. The second that you will hear about is to express CARs on non-alloreactive cells, such as NK cells, and Katie will present her exciting results, or monocytes. I think Sarah Gill is going to present those results. But the way that we've chosen is somewhat different. We've chosen to use T cells that retain their TCRs, but a TCR that is known to be non-alloreactive, specifically Epstein-Barr virus T cells. So we use the CD30 to prevent rejection and the EBV-STs to prevent graft versus host disease. And the reasons for choosing this course rather than the more common TCR depletion or separate platform is really partly pragmatic. We know that they're virus-specific. We've got about 30 years' experience using them effectively and safely, even in allogeneic settings, and partly because we believe that if you knock out the TCR in a significant with a significant number of CARs, not all, but a significant numbers, you reduce the activity of the T-cell, CAR T-cell in vivo. There are also definite positives in the sense that 90% of us have been infected with EBV. It's a persistent latent virus, so we're constantly being re-stimulated at low levels by EBV antigens, so that when we put these allogeneic cells in vivo, they should receive a low-level stimulation through their TCR, which may also help their survival. And last, but potentially not least, many CD30 positive malignancies are also EBV positive, so that you could get a dual hit both from the CAR and the TCR. So Carlos Ramos again took over this allogeneic study. Again, they're patients who've relapsed after usually multiple lines of therapy, again a median of 5, and we chose the line with the best HLA match. Now in reality, this turned out to be 1 to 3 antigens matching only. At 3 dose levels, 4 times 10 to the 7 3 times 4 times 10 to the 7 to 4 times 10 to the 8. And these are the 11 patients we've evaluated so far. 3 at dose level 1, 3 at dose level, sorry, 3 at, I think it's 4 at dose level 1. But if we look at the numbers of patients who are responding, it's only 1 patient completely responded at dose level 2. No patients responded at dose level 1, but 4 of 5 patients responded at dose level 3. And this we think is significant because this dose response curve implies that we're not looking at just the effects of flu-cy, but rather the benefits of the infused product. What about targeting tumor-specific antigens and overcoming the heterogeneity and plasticity? I think here the question comes down to how far can CARs alone take us? So CARs represent, as we know, 30 years of intelligent design, but TCRs really represent 300 million years of evolution and natural selection. And as scientists, of course, we're great believers in intelligent design, but I do think that natural selection and evolution are pretty important as well. And as Chiara pointed out, these cells, these receptors, TCRs and CARs, do function in a quite different way in terms of the antigens that they recognize. TCRs recognizing processed antigen, usually protein from within the cell presented on the cell surface in association with HLA, CARs recognizing mainly intact molecules on the cell surface. And as Carl Peggs will point out, I think, most of the desirable tumor-specific targets in solid tumors are internal antigens that need processing and presenting on the cell surface. But there's another advantage to TCRs over CARs that I think is somewhat neglected, and that is that TCRs interact in a different way with professional antigen-presenting cells than do CARs. So when a TCR that recognizes a tumor-associated antigen encounters an antigen-presenting cell in the tumor or an adjacent lymph node, it reacts with that APC. It generates cytokines and chemokines, the two of them together generate cytokines and chemokines that attract other T cells and other effector cells into the locale that will recognize any other tumor-associated antigens present on that APC or an adjacent one. And they too will be recruited and will recruit other cells in turn, so that you get this phenomenon of antigen cascade or epitope spreading. And the importance of antigen spreading, cascading, is shown very clearly in a number of studies. And one of our own studies that showed this was in patients with Epstein-Barr virus-associated malignancies. Now, these patients were treated with Epstein-Barr virus-specific T-cells, so we're working through the TCR, not through a CAR. And of the 23 patients with advanced relapse disease, 12 had a complete response. And I believe that all but one of them have remained in remission since treatment, which is different to the pattern we see with CAR T-cells. And one of the reasons we think this difference occurs is because if you look at the responses in the patient's peripheral blood, what you see is a rise in Epstein-Barr virus-specific T-cells, which is what you'd expect. But you also see a rise in T-cells reacting to other tumor-associated antigens that were not administered to the patients. This is an endogenous expanded response. These are important because if we take a tumor that is EBV negative and treat it directly with T-cells that are targeted to these same tumor-associated antigens, then we can get a significant response rate. Not quite as good, but 6 of 15 patients have a complete response. In other words, this epitope spreading response does contribute to the overall response and may benefit patients in terms of overcoming tumor heterogeneity and plasticity. By contrast, CARs recognize antigens directly on the tumor cell. Now they kill the tumor cell, they release antigens, those antigens go and do produce an epitope spread immune response. But we've looked very hard, and the only epitope spreading we can find is epitope spreading for antibody responses. Now it's true that those antibodies do include antibodies to proteins that are important in invasion and metastasis, but we've never been able to discover cytotoxic T cells from CAR T cells yet, and we haven't as yet been able to replicate any of the results by administering these antibodies to patients. So I don't want to say, however, that TCRs are the way to go for solid tumors, even though they recognize processed internal antigens and recruit new specificities. because CARs can do things that TCRs can't. They are MHC unrestricted, so it's much easier to come up with bank products and improve this accessibility issue. And secondly, and again somewhat neglected, but I think going to be coming much more to the forefront over the next few years, is that CARs can be designed so that they are very effective at discerning differences in the tumor and tumor stromal glycome, for example, the propensity to form multiple high mannose residues. And that means not just using scFv, not just using monoclonal antibody-directed binders, but also using other things that are also good at detecting abnormal glycomes, such as lectins. So what I think will happen is that we'll end up using TCRs and CARs, or TCRs that are more like CARs, or CARs that are more like TCRs, in the sense that they share these desirable properties. What about the inhibitory tumor environment? Well, for safety reasons and efficacy reasons, all immune effectors have a whole series of events that they have to pass through and in order to produce the optimal sustained effect. For T cells, they're conventionally divided into three sets of signals. Signal one is the antigen-presenting, the antigen-specific signal, whether mediated through TCR or CAR. Signal two is a whole sequence of checkpoint or co-stimulatory molecules which determine go, no-go phenomena. And then signal three, briefly, are usually cytokines, either membrane-bound or soluble, which determine how large the response is going to be, how much the clone is going to expand, how long it will last for, and also determine to some extent its polarization, helper, subregulatory, or cytotoxic. It turns out that tumors have mechanisms for inhibiting every single one of these steps. They can block access to the antigen-specific receptors, they can express immune stimulatory negative checkpoint molecules, of course the whole basis for the antibody therapy, and they can also produce inhibitory cytokines. In addition, they have a very unpleasant metabolic environment that's hostile to T-cell and other cell immune effector cell survival and proliferation. Some people call that signal 4, but I think the main way they're working is by modulating signals 1, 2, and 3, so it's perhaps an unnecessary complexity. There's been efforts to overcome every one of these problems individually. We've heard about the possibility of gene editing to overcome the checkpoint inhibition. I think probably the majority of studies have focused on signal 3, and Rainier Brentjens will likely talk about some of these tomorrow, I think. We've also been studying this approach. The commonest is to use exogenous cytokine, well, the original, I guess, was to use exogenous cytokine administration, or to overexpress cytokine receptors, or to have cellular secretion or tethering of the cytokines. And these have all produced some benefit. But the problem is it's very difficult to get the right amount of cytokine produced in the right place at the right time without causing toxicities. An alternative is to focus not so much on stimulation but on blocking inhibition. So by having a receptor that is a dominant negative receptor, such as a dominant negative TGF-beta receptor, beta receptor, which prevents inhibitory cytokine TGF-beta from acting, or to have an inversion receptor where you have an exodomain that recognizes an inhibitory cytokine, such as IL-4, but has an internal signaling domain derived, for example, from a gamma interferon. The limitation of this, of course, is it requires the tumor to be producing these inhibitory cytokines, and quite often when you start to use them, you find the tumor switches them off. So we focused on, or focusing now anyway, on constitutively active receptors, which are a kind of self-contained system. And the one I'm just going to briefly talk about is the IL-7 receptor, which is a heterodimer that induces STAT5 signaling when it's formed. And this is responsible for increasing proliferation of T cells predominantly, but also acts to upregulate anti-apoptotic molecules. And the main importance of IL-7 from the perspective of a CAR T cell is that it's one of the cytokines that's responsible for homeostasis. It's one of the cytokines that's produced along with IL-15 and perhaps others when we give lymphodepletion prior to administering CAR T-cells. So we thought it's possible that if we constitutively express this, we could do away with the need for lymphodepletion. So the constitutively active IL-7 receptor C7R constitutively activates STAT5 and CD4 and CD8 cells. It upregulates some anti-apoptotic molecules and downregulates pro-apoptotic. And when you add it to cells, T cells that have been stimulated by antigen and a positive co-stimulatory signal, you see there's a big increase in expansion, which is due to increased proliferation and reduced cell death, but you do not see autonomous growth. So the clone does gradually die back. It just takes longer to do so. And that effect is replicated in a whole range of different animal models. I'll just show a neuroblastoma model in which we target the neuroblastoma with GD2. If the T cell expresses both a GD2-directed anti-tumor CAR and the C7R, then the T cells expand much more than if it's just expressing the chimeric antigen receptor, and there's a corresponding improvement in the anti-tumor activity compared to the CAR alone. What happens in humans? Well, we're in the midst of a phase 1 clinical study of this approach, which is in neuroblastoma and osteosarcoma patients. 3 patients per dose level, 1 times 10 to the 7 to 1 times 10 to the 8. If we look at what their expansion is like, then we do see a very dramatic difference. When we used to use GD2 CAR, we tried it with and without lymphodepletion. And without lymphodepletion, the expansion was invariably 1 log or more less than the expansion with lymphodepletion. And that was a disappointment, but it wasn't a surprise because that's what you see essentially with other CARs. If you have the GD2 CAR, the same CAR, plus C7R, however, then so far, at least up to dose level 2, we've seen no difference in the expansion, no benefit from adding in lymphodepletion. Now, if this holds, then it would mean that we could give them multiple doses we're likely to need without having to give multiple doses of lymphodepletion, which would be a big advance for solid tumors in which, as I said, many of the patients are likely to be older and much less tolerant of lymphodepletion, at least in sequence. But if you go back to the tumor immune defenses, it's a pretty daunting array of possibilities that are open to the tumor. And as I said, they're responsive in the sense that they can turn off or turn on different mechanisms according to what is attacking them. And that means that while individual mechanisms, individual countermeasures may be valuable, in the end, I think we're also going to have to disrupt the underlying tumor microenvironment, degrade it to an extent that it really can't respond. And one of the ways of doing this, of course, is with oncolytic viruses. Our thought is that if you could combine an oncolytic virus with a cellular immune therapy, you might be able to get a synergistic benefit, because you could put into the oncolytic virus a series of genes such as chemokines, cytokines, checkpoint blockers that would then alter, that would then modulate the tumor environment, not just destroy it, but make it more like a virus, a hot virus infection rather than a cold tumor. The problem with doing this with a kind of conventional adenoviral or other oncolytic viral vector is that in general, the more genes you put into these oncolytic viruses, the more immunomodulatory genes you put in, the worse the oncolytic titer. So you may gain on one hand but lose on the other. Even if you retain the oncolytic titer, then in many ways it's self-defeating because you're going to be destroying the very cells that are producing the immunostimulatory growth and other factors that you want. So our approach has been a bit different. It's to make a kind of binary system consisting of an oncolytic and a helper virus. The oncolytic destroys tumor, but also replicates the helper virus. The helper virus contains the immunostimulatory genes that we're interested in. As that is produced and goes into the local tumor cells, both malignant and tumor stromal cells, it doesn't destroy them, but instead continues to secrete these immunostimulatory genes. And in, I think, 4 or 5 different animal models, this combination of an oncolytic and helper ad, a CAD-VEC, has proved very potent, even when it's only got interleukin-12 and PD-L1 without a chemokine in it. If we combine them, for example, with HER2 CAR T-cells and use them in a HER2 established orthotopic tumor model with metastases, we see some very striking results, which is that if you just give the tumor to these xenograft models, they die from primary and metastatic disease. If you give them CAR T-cells, they can control the metastatic disease, but they can't get into the primary tumor sufficiently well to eradicate it. Conversely, if you just give the CAD-VEC, you can control the primary tumor, but the mice die from metastatic disease because you haven't generated enough of an immune response. Only if you use the combination of the CAD-VEC and the CAR T-cells do you get the control of both primary and metastatic disease and the survival of the mice. So we're now in a clinical trial with this approach. We were very constrained by the FDA in terms of what we gave and when we gave it. We had to start with the dose of oncolytic virus without HER2 CAR T-cells. And we had to start with a dose of oncolytic virus that is actually about 100-fold lower than the dose that's generally given, because it's a 5 times 10 to the 9 total oncolytic plus helper, so 2.5 times 10 to the 9 viral particles. We've just finished dose level 2 and have just embarked on the first combination therapy. We treated our first patient about 2 weeks ago, and our second patient will be 3 weeks ago, and our second patient will be treated next week. But I don't have any results to show from that combination. So let me summarize what I've been saying. The impediments to immune effector cell function, treatment of solid malignancy, are quite extensive. They include accessibility of the product. These are being overcome for both autologous and off-the-shelf products. They include the need to overcome the issues with tumor specificity, heterogeneity, and plasticity, and a combination of TCRs and CARs or a hybrid TCR-CAR may help there. And we need to have potent tumor microenvironment countermeasures, either specific or general, or preferably a combination of both. So I think that the single point I really wanted to make from all these different comments is that solid tumors will require immune effector cells that function in a sustained and diverse way. And that's not going to work if we just try and keep going for one cell, one receptor, one time. We have to start thinking early of how we're going to combine these things. And that, in turn, is going to require us to have considerable modifications to regulatory and commercial frameworks that currently exist. Otherwise, it's going to take us a very long time, and I fear that we will run out of resources long before we make the impact that we could. So I'll just conclude by thanking the many, many people, a very small proportion of whose work I've very superficially alluded to. I'd like to thank our funders and, of course, our patients and their families. Thank you.